So let's just get started. We have more and more audience to join us. So today it's my great pleasure to have some of the leaders in this field in the synthetic lethality join us to discuss some of the hottest topics in this field. First, let me sort of introduce the BioSpark group for some of you may not familiar with. So BioSpark is a biotech-focused non-profit organization. So our vision is to establish a biotechnology ecosystem with collaborative and supportive scholar network, promote leadership and entrepreneurship, and facilitate the translation of life science. So apparently we have a lot of members and from top research institutes, biopharma companies, VC consulting, law, and CPA firms. As you probably can see, we actually have very diverse incomes and outcomes, and particularly we promote entrepreneurship, leadership, career development, scientific discovery translations, and scientific business collaborations. As many of you probably already joined us for our previous events, we have multiple events previously talking lots of biotech related topics here. And feel free to follow us on both Twitter and LinkedIn, and of course, events calendar. So here I also attach the WeChat group channel, and feel free to take the screenshot and join us. So today's topic is about how synthetic lethality powers cancer therapy. So the event has been organized by Devin Chen, Yisan Xu, and Qing Hong Yan, and without them, we couldn't make this. And also we got the extra help from Frank Ye on the event record and summary. So as I probably mentioned already, today is our great honor to have some of the leaders in this field to join us to discuss. So particularly we have the 4 speakers join us. So let me start to introduce Tuvik at the beginning, at the first. So Tuvik Becker holds a PhD in machine learning from the Hebrew University and has spent the last 20 years building and leading companies that apply machine learning techniques in a variety of domains, including aerospace, fintech, and primarily health tech. He's also a co-founder of MedAware, a leader in patient safety solutions, and has been serving as its executive chairman for the past 7 years. So he's currently the CEO of Pangea Therapeutics, a startup company employing genome-scale multi-omics to uncover clinically significant synthetic lethality and synthetic rescue interactions. So Pangea uses this technology platform to improve precision oncology through better prediction of response and resistance to treatment with existing and novel drugs. So welcome, Tuvik, to join us.
Thank you.
Yeah, thanks. Secondly, I would introduce Alan Huang. So Alan Huang is the Chief Scientific Officer of Tango Therapeutics. So he joined Third Rock Ventures as a consultant in 2016, where he played a leading role in the creation, launch, and build of Tango. So with multiple rounds of successful financings, strategic collaborations through the last few years, and eventually the IPO in April of this year. So he's also a scientific advisor for multiple US and China-based biotech companies and venture capital funds, including EQRX, Casting Capital, and Ligna Therapeutics. So Alan had more than 20 years of experience in oncology translational research, target discovery, and drug development, most recently from Novartis Institute for Biomedical Research as global head of oncology translational research. So under his leadership, the group advanced many key Novartis pipeline compounds. Most notably, he pioneered the pooled-based shRNA screening technology, which became the foundation for large-scale synthetic lethality discovery platform. Alan was trained as a molecular geneticist and had a PhD degree in biochemistry and molecular biology. So welcome, Alan, to join us.
Thank you for the nice introduction.
Yeah, thanks. And next we have Ethan Ropping joining us. Ethan Ropping is MD PhD in computer science. And he has served as a professor of computer science medicine in Tel Aviv University since 1995. Conducting computational multidisciplinary research spanning a wide variety of topics, including neuroscience, machine learning, and systems biology. So in 2018, he moved to the NCI, where he is chief of his cancer data science lab. So his current research is focused on developing new computational approaches for advancing synthetic lethality-based precision oncology and immunotherapy. So he is a co-founder of a few startup companies involved in precision oncology and cancer drug discovery. So including Pangea Therapeutics, from which he had divested and served as a non-paid scientific advisor and a fellow of the International Society of Computational Biology. So welcome, Ethan.
Thank you. Thank you so much for the kind introduction.
Yeah, thanks, Ethan. And next we have Yulian Zhang. I have honor to have Yulian Zhang to join us. So Yulian Zhang, PhD MBA, currently is a executive director of chemistry research evaluation as Fusion BioVenture. So Yulian is a highly accomplished leader with more than 20 years of experience across R&D and business development in large pharma and small biotech, Callen, Actinium, Blueprint Medicine, Lexicon, Vertex, and GSK. So she has significant expertise in medicinal chemistry, resulting in 2 FDA-approved drugs and 3 in clinical trials. She has PhD in organic chemistry from Temple University and an MBA from Stern Business School, NYU. So she's author of 15 papers and 2 books, chapters, and the inventor of 15 patents. Welcome, Yulian, to join us. Great. So finally, I think I should probably also introduce myself. For many of you may not know me very well. So I'm a bioinformatics scientist at Arcus Biosciences. So where I have been supporting multiple bioinformatics of multiple immuno-oncology drug development. I got my PhD from bioinformatics program at UCLA. And also I'm a serious entrepreneur and the founder of CellMaps and TopLab. So I've been working on genetic context-based drug response prediction for a while, and therefore today it's really my honor to have the chance to invite the leaders in this field to join us to discuss how synthetic lethality powers cancer therapy. That's great. I guess for many of you may already know some of the speakers. So let's just have our first section, which is about talking about some of the background about synthetic lethality. For some of you may not very familiar with this field. So as beginning, we are looking for Alan to briefly talk about what is synthetic lethality and how we can make oncology drugs based on synthetic lethality. Yeah, thank you, Alan.
Yeah, sure. I think, I'm going to talk about some background probably everyone in the field knows. So bear with me if you already know those knowledge. But for the people who are not familiar, synthetic lethality is actually not a particularly new concept. It was discovered in the year of actually, 1920s. That's when we don't know anything about our genes. Was discovered by a bunch of Drosophila biologists or geneticists. So what happened is they actually find some of the mutants that they can only recognize based on their phenotype. For example, the shape of the eye, right? 2 of the mutants, one is called glass, the other is called bar. essentially is describing how their eye looks like. Both of those mutants are viable. However, the scientists find that when you cross them, you're not going to get any viable progeny. So I think that gives the idea that each of the mutation alone can be tolerated, but combining them is not. I think that's the beginning of the idea about synthetic lethality. And post that, I think the group that make most contribution in this field are actually the East geneticists, because those are the first group that they can actually do those type of large genetic screen. This is well before we have CRISPR, shRNA, and be able to manipulate and engineer the human cancer cell lines. So I think, but the real concept of synthetic lethality and its application in treating human cancers, really happened in the year of 1990s. So I think Hartwell and Bill Kaelin each independently have brought up this idea of using this mechanism as a way of finding novel cancer therapies. I think the basic concept there is that synthetic lethality is a treatment or is a way of killing some kind of cells when there is a structural difference, right? And what cells has more structural and predictable structural difference than the cancer cell? So therefore, if you can find some unique vulnerability in those cancer cells with a known genetic defect or the structure change, I think that's a way that you can selectively kill the cancer cell. I just want to expand maybe a little bit. I think this idea actually, brought forward two concepts. I think it was at that time definitely very much novel and cutting edge, right? So one is about this therapeutic window idea, where essentially, the synthetic lethal interaction, because it only happens when certain genetic defect is there, suggesting the toxicity to the normal cells that doesn't have that defect is very low. Therefore, you can really separate the two. I think this concept that, just give you a little bit my personal experience, was not easy to establish. I think in the year of 2000, when I started at Novartis, building a functional genomics group using, at that time, well-based shRNA screen, right? So I remember we had a lot of debate about whether BRAF, which only kills 1 or 2 cells, versus PLK1, which kills almost every cell, which one will be a better cancer target, right? So I think 20 years later, that's probably not a debate anymore. But I think that just give you a flavor. This concept of the therapeutic window is an important one, and it takes some time to adopt. And this is what built in for the synthetic lethality interaction. The other piece I think from this concept brought forward is patient selection, because that's a building concept as well, right? You need a target, and also at the same time, you need a context that fit into this target. This context is patient selection strategy, essentially, which becomes so important. I think, if you thinking broadly about all the cancer therapies, most of them are oncogene addiction target, like all the RTKs, kinases. I think the idea there, to be able to select the patient and also be able to have a clinical proof of concept in 20 patients, right? If you have a BRAF inhibitor, you select 20 BRAF mutant melanoma patient, and if you don't see response, the drug is not going to work, right? So I think unlike some of the drugs that people have been developing lately, where no patient selection strategy is out there, right? You have to run those large phase 3 trials. After 1,000 patients, it doesn't work, right? So I just think, even from the practical point of view, to have a patient selection strategy, particularly in this case, a built-in strategy, is actually super important. So yeah, maybe I diverse a little bit too much. So on the path of patient, the synthetic lethality, you know, I believe this is in 2005. This is when Alan Ashworth, who is also a founder of Tango, discovered in the laboratory that using a PARP inhibitor and using a pair of isogenic BRCA mutant versus wild type model. This is during the year when genetic engineering and getting those isogenic are still very challenging, right? So he has demonstrated that he saw like, you know, 1,000-fold difference of sensitivity between the BRCA mutant and the wild type cell line. So that was very striking. I think this is the first example where it actually saw this concept, you know, played out. And clearly now, this is also the first concept that we are seeing clinical, you know, approval, right? So that's almost, I view this as a proof of concept for the entire synthetic lethal field, right? And then since then, I think, you know, with the development of the genetic tools and also the model system, particularly large project like Cancer Cell Line Encyclopedia project, as well as the large screen project like the Drive screen from Novartis and also the Achilles screen from Broad, has really, you know, powered forward this field, you know, very dramatically. I think, you know, there are many more interaction being discovered, such as, you know, ARID 1A, 1B, SMARCA A2, A4, you know, MSH and Warner, and PMT5, MTAP, et cetera, et cetera. So I think there are, you know, a lot more to be discovered. You know, some of them might not be discoverable just using the large cell line panel, and some of them you might need using a different readout. But I do think that that's fundamentally a very important paradigm. You know, and also if you expanded this concept, you know, a little bit, I think almost all cancer therapies, you will need a therapeutic window. You will probably need a patient selection strategy. You know, it's hard to imagine anything just cure all the cancer types. So in that bigger context, I think this paradigm will play out even more in general. So hopefully that gives a little bit background of, from my angle, how the synthetic lethality as a concept plays out.
Well, thanks a lot for Alan to share this insightful, high-level introduction for synthetic lethality. It's very nice. So next, we are going to have Ethan to also talk about the computational-based approach to detect those synthetic lethality partners, and also how that's gonna help us on precision oncology.
Sure. So before that, I want to say that synthetic lethality, in my mind, has two main applications. Okay, one of them, as Alan has.
I think we need to.
Sorry for the disruption. Yeah.
No worries. So one of them, as Alan has so beautifully described, is drug discovery. And as he described, the big advantages of the therapeutic window and the ability to select patients, right? Biomarkers of therapy, which is, of course, a holy grail in modern drug discovery and treatment. But the other application is precision oncology, okay? So imagine that you have a set of targeted and immunotherapy drugs in an arsenal, and comes in a patient, and you want to choose the best drug that will most effectively help the patient from an existing arsenal, right? So this is the scenario of precision oncology. Now, just imagine that you would know the synthetic lethal pairs of each of these 20 or 30 targeted and checkpoint therapy drugs, right? So now, given a transcriptomics, copy number, methylation information from the patient's biopsy of the patient's tumor, you can estimate for each drug what is the state of the synthetic lethal partners of the targets of the drug. So for each patient, you can score 20 or 30 targeted immunotherapy drugs. You can score the likelihood that they will help the patient by the state of inactivation of the synthetic lethal partners of each drug. Okay? Obviously, the more synthetic lethal partners that are inactivated for a given drug in a given patient, the more likely he is to respond, at least by synthetic lethal mechanisms. Okay? So the multi-billion-dollar question is really, how well can you do that? How well can we identify the synthetic lethal partners of our current cancer drugs, or even non-cancer drugs, if you want to go wild in your imagination? So I hope this application is clear before I go and describe the intuition of the black magic of how we aim to do that. Is it clear? Is it okay? Well, whatever. I'll assume it's okay. Okay. So over the last 7 or 8 years, my lab has developed an approach to try and do that, to try and identify the synthetic lethal partners of each and every targeted and immunotherapy drug. The way we do it is we analyze a large cohort of patients' data that, let's say, has 10,000 tumors like the TCGA, and we aim to identify what we call pan-cancer interactions, synthetic lethal interactions that are shared, are a backbone of many cancer cells. Of course, we know that synthetic lethality is context dependent and so on, but for various statistical power reasons, for now, we developed this approach. And the intuition, I can really explain to you the intuition of what we do in 1 minute, and it takes us years. So either we are complete idiots or the situation is a bit more complex. So, but I'll explain the intuition. Suppose 2 genes A and B are synthetic lethal, and suppose you look at the state of inactivation, their mutations, their copy number loss, their low transcription, their methylation of the promoters, what have you, in each of these 10,000 tumors, okay? And suppose you find pairs that are, while they are inactivated, each of them alone, they are not inactivated together. That may support evidence that they are synthetic lethal, because whenever they were inactivated together, they are reduced the fitness of the cells, and you don't see them in the samples of the tumors, of the 10,000 tumors that you see. So that's one intuition. It's important to note that we don't start just, from the blank. We analyze, as Alan said, DepMap and CCLE and whatever, and there's some intuitions by which you can, from that analysis, start with a pool of potential synthetic lethal candidates of the different drugs, okay, that arise from these in vitro screening pools. But typically you can get hundreds of thousands of such candidates. So we take this initial pool, we go to the patient's tumors, and we aim to identify the subset which is supported by the clinical data. So as I said, we are looking for pairs that are not inactivated together, okay, because they are then selected against if they are truly synthetic lethal. And even when they are inactivated together, we are actually looking for such pairs where the survival of the patients is better than expected, because that again may testify that they are synthetic lethal or synthetic sick, because they reduce the fitness of the tumors and the survival of the patients is better. And finally, we employ some evolutionary phylogenetic analysis, because as Alan said, there is a lot of literature across species that shows that synthetic lethal pairs tend to occur between functionally related genes, which have a shared phylogenetic history, and so on and so on. So lo and behold, we take hundreds of thousands of initial candidates which emerge from this in vitro analysis, and we aim to identify a small subset for which there is clinical evidence that it may actually occur in the patients. And then we use these as predicted synthetic lethal partners of the cancer drugs. And we can show that for many of the targeted therapies and the checkpoint therapy, we get reasonable predictive power of the patient's response to these therapies, which enables us, we think, to devise biomarkers and to think about whole exome transcriptomics-based new generation of precision oncology. And this is something that we are now actually bringing to clinical testing and moving to clinical trials in the NCI. And I think we are fortunate to have in the audience my close collaborator and friend, Ken Aldape, who is chief of pathology in the NCI. And together, and of course, with the support of CCR leadership and many fantastic clinicians, we are embarking on an initiative to sequence hundreds of patients in our center, both for whole exome sequencing, RNA sequencing, and methylation. And together with top clinicians in the NCI, gradually and carefully study our ability to help patients, to treat patients with this new generation of synthetic lethality-based whole exome precision oncology. Of course, all the evidence that we have provided up until now had been retrospective. Now we are going to study it prospectively, and it remains to be seen. I can just promise that we will try, we will do our poor best, and fingers crossed that we may perhaps succeed a bit. So.
Great, thanks Ethan a lot for this. For myself, I'm also a computational biologist, so I'm very excited about this and definitely looking forward to hear more from you and also from this field. So next, I'm going to actually stop sharing of myself and enable Yulian to share her screen to talk about some of the business side about synthetic lethality, investment, and therapeutics companies. Welcome, Yulian. Yeah.
Sure. Thank you, Ning, and thank you, Alan and Ethan, for such a great fundamental introduction to synthetic lethality that saves me a lot of time. So hello everybody, and thank you being here for on the weekend, and thanks for the organizers' invitation. So in the next 5 minutes, I will just briefly cover these topics: approved PARP inhibitors, synthetic lethality drugs in the pipeline, synthetic lethality business deals, and finally fundings for these 3 leading synthetic lethality companies, including Adiana, Tango, that's where Alan is working, and the Repair Therapeutics. So currently there are 4 FDA-approved PARP inhibitors, including olaparib, rucaparib, niraparib, and talazoparib. And in addition, there's 1 approved PARP inhibitor by NMPA in China called fluenzoparib, discovered by Hengrui. The first FDA-approved PARP inhibitor was olaparib. And as you can see, last year's sales was about 1.8 billion. The forecasting sales for 2025 is about 6.8 billion. Encouraged by a huge success of PARP inhibitors, a lot of effort has been put into the field since 2013, resulted in multiple drugs in the clinical trials. So at least 18 drugs other than PARP inhibitors are in phase 1 and 2, inhibiting 8 different targets: DNA PK, ATR, PKMYT1, MED2A, CHEK1, V1, MYC, and RED51. In terms of collaborations, this slide summarized representative and important deals in the past 7 years. Large pharmas such as AstraZeneca and Merck spearheaded in the field since 2013. In 2017, small biotechs such as IDEAYA started the collaborations with institutes. A few years later, more big pharmas realized the potential of synthetic lethality and decided to enter the field quickly by striking deals with small biotechs. In 2020, last year, there are 3 significant deals between Gilead and Tango, GSK and IDEAYA, BMS and Repair. The deals are pretty significant. The upfront ranges from 50 million to 145 million, even for the preclinical discovery stage programs. Next, I will touch upon these 3 companies. First, IDEAYA started 2016 with 48 million Series A. 2 years later, they secured 94 million Series B. In 2019, they went public with proceeds of 57.5 million. As of yesterday, the market cap is about 661 million. So here is their pipeline. The most advanced program, MED2A, is in phase 1. Tango Therapeutics just went public by a non-traditional SPAC merger in April, less than 2 months ago, with proceeds 353 million. Tango started in 2017 with 50 million Series A. 3 years later, they raised 60 million Series B. Here is their pipeline. Alan can speak more to that. And the most advanced program, PRMT5, is in the IND enabling stage. So 3 months later, after Tango, Repair Therapeutics started with 68 million Series A. And 2 years later, again, they raised 82.5 million Series B. Last year, they went public with 253 million proceeds. As of yesterday, the market cap is about 1.15 billion. Currently, they have 2 assets in the earlier clinical trial stage, ATR inhibitor and PKMYT1 inhibitor. With that, I would hand back to our moderator, Ning, and I'm sure we will have more discussions during the panel session. In the meantime, you are welcome to fit yourself into one of these categories on this picture. And thank you very much.
Great. Thanks a lot for you, Dan. That's very, very nice presentation. And I really love the business side about the synthetic lethality companies. And I think as many of us in the industry, we all care about the, what are the current pipelines for those therapeutic companies, and how about their financial side about those companies, like how big they are, like how they're gonna grow up to some point. So I think that's very, very nice overview of this. Of the industry of those companies. I think our audience must already have some sort of sense about this field, both in scientific side and the business side. So next, I think we're all eager to get started about the panel discussion, about the questions we have collected from our audience. So let me just start the questions. And for the audience, I think Devin already mentioned, like if you have any questions or if you want to interact with speakers, and just feel free to put that into the chat box or raise your hands and I will let you in. So let me start my first question here, which is, I think this is my favorite one. This is my personal favorite one, which is like we all care about, like what are the current strategies of synthetic lethality powered cancer therapeutics companies. I think Alan and Tuvik might have ideas on this. So I would just let you guys start. Yeah.
Alan, would you like to go?
Sorry,
you go ahead.
Okay.
Oh,
So at Pangea, our approach is a computational approach. Basically, we are following in the large scheme that Eitan has described before. And I would say that there are three main premises on which our strategy is focused. The first is we try to look at network effects rather than just single synthetic lethal pairs. So we find that it's much more effective if you have a target with multiple different synthetic lethal partners that are inactivated in the patient tumor. That may induce a much stronger effect than when you solely rely on one pair. The other basic premise is we use extensive analysis of preclinical and clinical data to complement the in vitro screens and in deriving what interactions are really clinically significant synthetic lethal interactions. And the third premise is we focus on transcriptomics as the basic source of knowledge about the state of activation of the genes, and we complement that with data coming from copy number variation, other variant calling, and also methylation data from promoter methylation. So based on all of these, we try to really build the network of synthetic lethal interactions, and that allows us to drive both novel drug target discovery and, as Eitan mentioned, the ability to better predict response to treatment with a variety of targeted therapies that are already on the market, and find for each patient which therapies are more likely to result in response and where there's less likely to be resistance to the treatment. So different applications, and we'll probably talk about that more. So Helen, please.
Yeah. So for us, I think, so first of all, thank you for Yulian for giving a comprehensive review and a comparison. I think it's very interesting to see that. There are probably a few other players. I think the field right now is much more focused on the DNA damage repair, right? So almost everything that Yulian has listed are there. I think that's there for a reason, right? Those are where the success has happened. Probably people felt those are the lower hanging fruit that can be grabbed easily. But I think those are certainly important. We are working on DDR targets as well. But I think if you're looking at for the future targets, there are, even for tumor suppressors, 600 of them, right? So there are many flavors of tumor suppressor genes. I think worthwhile to really generate some studies to try to identify novel synthetic lethal targets, right? And many of them, I think even though now the Achilles screen, that map has really grown a lot with 600, 700 cell lines. I think there are certain intrinsic limitations for those tools. So in our case, we think you still need to complement a clean system with a real world system, which is the cancer cell line encyclopedia. The clean system meaning engineered system that you can combine them and using orthogonal ways and even computational ways and others, looking at patient data to be able to narrow it down to something that is real and strong with a really strong phenotype. So I think methodology wise, I think those are the things. And there's many other tumor suppressor genes other than the DDR field, right? And on top of that, I think also we felt like, you know, the phenotype for many years has been really narrowed down to a, you know, kill the cancer cell on the culture dish, right? Which is, I think, definitely works well for PARP inhibitor and everything. But I think the last 10 years of the success of immune oncology at least tells us one more thing, right? You can kill tumor cells in a different way, right? You know, basically to trigger the immune cell to kill it, right? So, and if you put, for example, that concept into the thinking, you can actually include that as part of your synthetic lethal equation, which just means that, you know, if you have a certain genetic defect, then, you know, targeting something that you know, may trigger the immune system to really kill that particular cancer cell rather than a direct cell autonomous effect. So I think that concept, you know, was really, I think, very interesting to me as a cancer biologist, because I, you know, in the early days interacting with immunologists, I find we were like, you know, people from different planets, right? They never care about cancer genetics, you know, T cell, all these immune-based cells are the things that they really care about. So I think there are definitely some interaction, right? You can imagine, like, for cancer cells, they have all these mutations. They're supposed to be recognized by the immune system. The reason they can't, you know, must be some sort of evasion system that they developed, and you know, that initial culprit might originate from the cancer cell itself, and might be some of the genetic changes that it carries, right? So, but if you think about that way, right, then you can expand the whole thinking about the synthetic lethality into immune-based kill as well, right? So obviously you're now increasing the complexity of the system, where especially if you're using a model system, you know, in this case, we have been using quite a bit of in vivo screen, which for people who have done those, you know, those are very challenging to do. But, you know, I think the technology has really advanced a lot. So to be able to capture some of the nuances and in combination with some of the cell line tools, I think this is, to me, a very exciting field. But obviously, immune-based phenotype is just one vineyard, right? So you can imagine, you know, thinking about different output. Essentially, you could really generate different flavor of synthetic lethality. Yeah, so those are the areas that I think, you know, we are doing lots of the traditional hardcore synthetic lethal screens and explorations. But I think on top of that, just to think more creatively in terms of the, you know, the context as well as the phenotype is another area where we think are very exciting. And just for Yulian's point, the whole collaboration we had with Gilead is really based on that immune-based screen, right? So that's really their major interest, and they see this as a potentially interesting angle for them to get into the immune oncology as well.
Great. I think that's really nice discussion from both Alan and Tuvik. I think let me summarize in this way. It seems like for the from Tuvik's side, it's not mostly important components about precision oncology, like how we can use computational approach to better detect the signals, and of course better patient stratification. And for Alan's side, it seems one of the trend or next wave is what Tango currently doing is like adding the immune component, which is transition from tumor intrinsic to immuno-oncology based approach, which I definitely think this should be the future. Let me just get one more setting for our Clubhouse audience. If you guys couldn't see us, like Zoom, like you can go for the Twitter account to follow us, for you to follow some of the speakers' bio and some of the screenshot. And next, I'm going to sort of have the following question on this is like, I also noticed that for both Alan and Tuvik mentioned, most of that focusing on the identify drug target. So then the natural question from the audience that after identify the drug target, how the target discovery therapeutics companies can compete in biology, chemistry, and clinical side. Like what are the strategies of things like after the drug target identified, like what can you guys do on that side? That's some of the curious side from the audience.
Vivek, you want to start?
Sure. So Pangea works with some drug discovery companies, and in our collaborations, there's emphasis on closing the loop between the lab and the clinic and the computational biology group. So when we identify a good target or a good biomarker for a particular target, we are going through a process where the biomarker guides the preclinical work. So experiments are designed based on the biomarker, and then data from these experiments feeds back and informs a biomarker design, and that's a continuous process of improvement. And later on in the clinical stage, you get additional data from actual patients, so you can improve that further. So things don't end when you identify a target; they just begin then, because we see it as a process. Biomarker-guided clinical trials are really way more efficient than classical ones. So being able to improve your biomarker based on real-world evidence is very strong. So there's need for this continuous loop of improvement in the biomarker.
From my side, I think, one thing I just want to emphasize, I think we actually do have a world-class drug discovery team, right? So I think, it's not like if you do target discovery, you cannot do drug discovery. So I think that's clear. We actually made, I think, the most leading product on the true MTAP selective PRMT5 inhibitor. I know many big farmers are trying to do that and wasn't able to get there. So I think that's a testament of, small companies, we can also build a world-class and drug discovery team. But I think just in terms of the general strategy, right? So, one thing we are trying to avoid is to get too fancy with chemistry, with drug discovery. Meaning, usually you think about companies in two flavors. One is to dig more in biology, the other in technology, right? So you have companies who are working on drugging the undruggable. You have companies that working on, to really dig up novel targets, novel biology, essentially. So I think to try to do both is probably not wise. So from the very beginning, our strategy has been avoiding, for example, protein-protein interaction or using siRNA to target something, right? So, it's not like we cannot do, it's really to make it more clearly focused, right? So I think this is helpful for any company to be clear, right? You cannot do everything. So that, I think that's one. So we do world-class conventional drug discovery. This is what I usually say. I think associated with that, which is interesting, we do see, target that we discovered, there are some different flavors. Obviously, difficult to drug target, we usually would just ignore. But there are targets that are probably, not so challenging for targeting, but it's completely novel. No one has tried. And in some cases, people will consider those high risk. But we had quite a few success examples where, just because no one has tried doesn't mean that's difficult, right? So we might be the first one tried it, and and it went very smoothly, right? So that's one class. There's another class which is even more interesting. Those are the targets that are out there, people may have worked on that for a while for different reasons, different biology, different reason, or even different indications. And but really, there's no path to go forward based on existing knowledge. But we find interesting new insight into those targets, and that those potentially could provide a chance to really jumpstart the program. I mean, again, if there's a approved phase 3 drug out there, that's obviously a different story. But many cases those are not, and because of the lack of the biology, I think those programs, they may have beautiful compound out there, but really has no path forward. So those, I think, could also be very interesting settings for us to take advantage. And we see both cases, right? Novel target, but druggable using traditional way, and other things that we be able to take advantage of some existing effort.
So can I pitch in, Ning?
Sure, go ahead, Ethan. Yeah.
Yeah, so yeah, I want to just bring examples of things that my lab has done in this space, in yet applying synthetic lethality to identify new treatments and synthetic lethality treatments and treatment opportunities. So around 2018, Silvio Gutkind, who is chair of pharmacology in UCSD and a friend and collaborator, came to me and told me about uveal melanoma. Maybe the audience knows it's a very aggressive form of melanoma, basically doesn't have any treatment. There were some inhibitors on the pathway, but they don't work. And uveal melanoma is, as Alan said, is caused by a major oncogenic event which drives the disease, and it's an activating mutation in a gene called GNAQ. And the idea was, that Silvio said that he challenged me with, was to look and search for synthetic lethal partners that are specifically synthetic lethal when GNAQ is activated. And that's what we did. We analyzed melanoma, uveal melanoma patients' tumors, and we identified a few such synthetic lethal partners specific to the activated GNAQ. And we found FAK to be a very strong one, and Silvio's lab did very deep mechanistic work to validate that and identify some mechanisms. And we could repurpose a drug to that. And now, we are running a big clinical trial to see how well it can help patients. The trial is still going, and we'll see, of course. So synthetic lethality offers a quite unique opportunity to target undruggable oncogenic drivers, because it's very difficult to target them directly. But not sure that you always can, but if you can find excuse me, synthetic lethal partners of these events, then you can get very interesting treatment opportunities. So not only tumor suppressors, but also driving oncogenic events are good targets. I didn't mean that to have such an effect of what I said, guys. I hope everyone is alive and kicking.
Great, yeah. I mean, it's really a nice discussion. I mean, it seems like different, even for just three of you have very different thoughts on the same things. I'm pretty sure the audience will learn very different things, like from their own side also. I think we already accumulated some of the questions from the audience, and maybe I found some of them are really interesting. Maybe we should just pick up one to just give them a chance to ask the speakers. So one of them is actually I think this is very interesting one, which is from Ji Yang Yu. So he or she is asking that how feasible to have FDA okay with using total RNA-seq derived synthetic lethal or biomarker score, or you will have to reduce to a small panel. Any successful examples of using whole transcriptomics to get the treatment? Thanks. I think that's might be some questions Ethan or Tuvik want to touch a little bit more.
Sure. Tuvik, do you want to go ahead? You want me? Whatever. Go ahead.
Whatever.
Go ahead.
So I really think it's a great question, and time will tell. I don't think anyone knows right now. I don't know of any existing example, but the question is whether we really need a whole transcriptome or whole transcriptome data in order to for the test itself. It's important during the discovery stage, but once you have your synthetic lethal based biomarker, you can probably reduce it down to a small panel and have very effective results, a cheaper test, and more reproducible data. So I'm not sure there would be a need in the near future to really go with whole transcriptome RNA-seq to the FDA approval stage. But maybe Eitan has a different perspective on that.
Yes, I do. I strongly believe that it will happen. It will happen, and it will happen in a scope of 5 years. Tumor transcriptomics will become a standard thing that oncologists will ask, and for patients. It's already growing up. Its use is growing up. Companies like Tempus and Caris, as you know, and so on. It's bound to happen because of a simple fact. Currently, the fraction of patients that benefit from the standard actionable mutation panels, for targeted therapy in precision oncology is quite low. It's, varies in the estimates around 10%, 15%, and so on and so on. And it's very clear that you need to look at the scope of tumor vulnerabilities and potential treatments across the whole exome, and you need to do whole exome sequencing and whole transcriptomics, and it will happen. It's bound to happen. Think about it. The cost of doing whole exome transcriptomics, RNA sequencing and methylation per patient now, depending on the depth and so on, is about $3,000, $4,000 per patient, okay? This is a trifle. It's nothing compared to the cost of treating cancer patients. The estimated cost, annual cost of treating cancer patients currently in the US is $150,000 to $200,000 a year, okay? So if you think about it, it's insane not to do it, provided that we can have indeed an approach to recommend therapies for patients based on the transcriptome. Let me tell you one thing. Even for our best established targeted therapies, let's say EGFR or BRAF targeting, as you know, there is a considerable variability in the response of patients. What we have shown in a paper that we recently published retrospectively, not prospectively, is that we can take such patients in clinical trials. All of them have a mutated EGFR or a mutated BRAF, and we can still, based on the expression of the synthetic lethal partners of EGFR or BRAF, based on the expression state of these predicted SL partners we give within a given patient, we can predict, again, far from perfect, but with overall around 75% accuracy, we can predict who will respond and who will not. And all of them have the target actionable mutation. So it's bound to happen. The writing is on the wall. Okay, that's what I think, but you know.
I hope you're right.
Well, it remains to be seen. I have a long history of being wrong on many occasions, so I wouldn't be too optimistic.
Yeah, no, I hope the large scale clinical trials that you're pushing for at the NCI might actually tilt the scale on that. So that may be a tipping point.
We'll see. Remains to be seen. But I think that irrespective of our efforts, the trend is certainly to incorporate transcriptomics and other things in bulk expression. By the way, I'm getting out of the scope of this discussion, but I think that in a few years, there will also be single cell transcriptomic used to guide precision oncology. The cost is 15K, 20K, but it will go down. And in my lab, and I'm sure that in many other, leading computational labs in the US and beyond, we are developing an approach to try and do that, to try and tailor therapies based on the single cell tumor transcriptomics. We will release our first paper on that on bioRxiv in a month or two. So stay tuned and see what one can do.
Great. I mean, certainly, I mean, I have heard back from the audience. Precision oncology is certainly one of our most interesting topic for this synthetic lethality. So it seems like, let me summarize what we have discussed, like the potential applications for synthetic lethality in precision oncology, including the synthetic lethality target discovery. This is the first one. Second, it seems like we can use that to identify the biomarkers. And third one is like we can predict patient response. It seems like those three are what we have talked a lot. But I think the audience also curious, like what are the other potential applications might also interesting for synthetic lethality applications if we miss any of them.
So I can answer, but unless maybe others want to, if not, I will pitch in.
Yeah, go ahead. Yeah.
So one obvious additional application is, of course, looking for synergistic combinations, right? And well, synthetic lethality is one natural way to search for synergistic combinations. So we just submitted a paper to sell. Of course, they didn't say if they're taking it or not. I think we'll put it in the bioRxiv next week. It will be on bioRxiv. So it's actually, Alan, you mentioned that you were in Novartis, right? So actually, it's a collaboration with Cyril Benes from MGH, who joined Novartis. It's like 3 years work together, charting the landscape of combinations in non-small cell lung cancer, a joint, very large experimental computational, I think, tour de force of combinations. And we're very interesting. Some of that is done by screens, and some of that is done by looking for synergistic combinations by synthetic lethality. And since the paper will be out next week, I will not say more, and you can look at it for yourselves.
That's great. I think that's definitely one of another very interesting topic in, at least I know in the pharma, which is how you combine different treatment drug together to generate a synergistic effect to better treat the cancer patients. So I think this is definitely a very good point. Like we are very looking forward for the paper.
Sorry, Ning, I just want to say we did already publish some synergistic synthetic lethal-based combinations. I just mentioned very briefly, one of them is with Zev Ronai in Nature Cell Biology in 2019 as a synthetic lethal synergistic combination in pancreatic cancer and in melanoma between MEK inhibitors and L-asparginase. And recently we published a paper in Science Advances with a group of CMR at the Weizmann Institute, where we predicted synergistic combinations in triple negative breast cancer, and they validated them experimentally. So, you know, sometimes if we are lucky, one can succeed. Sometimes we fail. You know, what we do is noisy and it's far from perfect. And perhaps the most important thing that I want to tell you guys before I forget or I won't have a chance is that we are developing computational approaches to identify clinically relevant synthetic lethal interactions. I think we have a signal and benefit, but we are certainly, you know, in early days or midway. Okay, there's still a lot that can be done to refine our computational approaches, and they will become much better in the next few years. We are doing a lot of research on that in my lab, probably maybe also other academic labs, and surely Pangea is also pushing the envelope. So what you see now is not what you'll see in 2-3 years. We are computational guys, so with some luck, we can move relatively fast.
Great. That's,
can I follow up on
go ahead, Alan. Yeah.
So yeah, no, maybe I can share my experimentalist point of view on that combination, right? So I totally agree. I think the way, again, when we talk about synthetic lethality, right, that if you look at all the classic chart, it's like gene A, you know, cross then leads to a gene B dependency, right? That has always been the way how this was depicted. But if you think about this logically, right, you know, why all these things are so you know, simple and straightforward, like a one-to-one relationship, right? Why couldn't it be you have multiple lesions that leads to a dependency of some gene, or you have a lesion that needs to target both gene A and B to be able to generate that synthetic lethal interaction, right? So I think that obviously we haven't even solved the one-to-one relationship yet, right? So this is incredibly complicated. I think, you know, I definitely believe the computational power could be the one to help us. But on the, you know, again, experimental side, we have been, Tango has been working really hard to build some of the tools to address those, what I call higher order synthetic lethal interaction. Mostly we're using those CRISPR tools that can take out more than one gene, sometimes 2 genes and 3 genes. I think those will be very useful, like to address the question, like, you know, for example, you can take down 2 tumor suppressor genes at a time and build a library like that and asking, you know, the sort of synthetic lethal question. Or you can, you know, take a library that targeting, for example, you know, I think the randomly 2 gene combination will be a gigantic number, right? But you can do a rational combination, for example, paralog and others, right? And some of the pathway hit nodes to be able to get you idea of this combination, you know, if you don't have a drug, right? If you have a drug for one of the nodes, then obviously you can do drug-drug combination or drug CRISPR combination type of things. But I do agree, I think this is one of the sweet spot rather than looking into one by one. You can use this type of screen and technology to looking for that.
So if I may pitch in, I think this relates to another question raised by the audience, tying synthetic lethality to whole arm loss events. So those higher order synthetic lethal interaction that you're talking about are particularly interesting in that context, because you have multiple losses of multiple gene losses in the arm loss, and then you can tie that 2, 3 or more gene effects to another gene targeted in a different chromosomal arm. So that's one particular case where such higher order events would be very interesting and compelling.
Great. I think we already discussed a lot, and I promise this is my last question. And also, I want to give the audience the chance to raise your hand, and we will unmute you to ask questions. So while I'm asking my last question, so if you have any questions, please raise your hands and we will let you in. So my last question is actually related with CRISPR technology. I think this definitely related to what we have discussed. And I think since I'm also working as a synthetic lethality, I definitely feel like CRISPR is one of the tools that can power the next wave of synthetic lethality. So then the question is like, how do you think about the CRISPR technology that could power the discovery of the synthetic lethality? How that gonna happen? I think we already touched some of them. I just want to see whether you guys can expand that a little bit more, unfold that a little bit more for us to fully understand how the CRISPR specifically can power the synthetic lethality-based drug discovery.
Yeah, maybe I can start. The technology itself, I think, for someone who has been working with shRNA for 10 years in the past, I think it's definitely a huge upgrade, right? So I think the precision of the technology itself is non-parallel, right? However, I do think this is a fast-growing area, and there are, different flavors of CRISPR technology that are emerging. I think I can, maybe raise a few, right? One is clearly even just between CRISPR-Cut and CRISPR-I. I think that the difference is quite big. We have screens, we have done parallel head-to-head comparison, and we do see there are quite a bit of differences that, later on we confirmed is really driven by the MOA of CRISPR-I and the CRISPR-Cut that can give you a different phenotype. And I talk about the combo CRISPR technology. I think there are also, the traditional Cas9 can do that. It's a bit more cumbersome, but the new enhanced Cas12a will probably help you to take down not only 1, 2, or 3 genes, right? So I think you can imagine a whole slew of implications for that particular technology. And the last one I think is also important is that, since I talk about the in vivo screen, right? Some of the technology involving enhancing the coverage or weeding out the real signal from the noise, right? Typically, the coverage has been an issue for in vivo screen. It is going to be very important. Like, for example, some of the barcoding system that you can use to basically add a level of confidence about your data. Obviously, those are not the core technology itself, but I think those are equally important.
Any comments from the other speakers?
I know nothing about this stuff, so I reserve my right to remain silent.
Great. Great. I mean, I saw one audience raise his or her hands. So while I'm let him in, I also want to see for the audience from Clubhouse. We also want to give a chance for them to ask questions. So also, like if you are in the Clubhouse, so feel free to raise your hands. Or I don't know whether you can raise your hands, but we certainly will find a way to let the audience from the Clubhouse also ask questions. So while the audience from Clubhouse thinking about their questions, like let's just let the Zuye let him to ask questions. Yeah, go ahead.
Thank you for sharing. It's very exciting. I have 2 small questions, and the first one is when we talk about the synthetic, so regarding to the PARP inhibitors, what I learned that is those kind of PARP inhibitor will very effectively to kill those kind of BRCA1, BRCA2 mutation patient or HDR deficient patients. So, but those kind of patient percentage in clinical actually is not that much high. So my first question is how to target those the rest? I mean, with HDR proficient or with the wild type BRCA1, BRCA2 wild type patient. And the second question is, which I learned is that even those kind of, I mean, in clinical, even those BRCA deficient BRCA mutation or HDR deficient patient, after those kind of PARP1 treatments, at the beginning, I think is effective, is quite effective, but later those kind of patients, the tumor will become quite resistant to those kind of PARP inhibitor treatments. Although you increase the dose, or it may not have such kind good effect. So from your view, I mean, how we overcome those kind of PARP resistant? Thank you.
Alan, do you want? I think you have the most experience with PARP inhibitors, maybe.
Sure. Yeah. I think for your first question, that is the paradigm for target therapy. You're going to have fragmented patient population, and even though Yulian just showed that that's like $6 billion sales, right, in 2025. So that's already a pretty big market, even though that's a fraction of the patients there, right? So, the rest of patients, I think, we'll need to dissect them based on their genetic context and, whatever the defect they have, and figure out treatment, right? One small fraction at a time, right? So that's how I view it. If you look at lung cancer, that's what happened, right? Initially with EGFR, then ALK, RAS, TRAC, all these things, even now even KRAS, right? This is eventually how we can figure out. So one step at a time. The rest of the HRD, wild type patient, they may have nothing to do with PARP inhibitor. We'll have to probably use additional strategies. The second question you asked about PARP resistance, I think that's very clear, right? So, for instance, the BRCA revertant, right? If the mutation happened in certain way that allow the tumor cell to reach recombinant their BRCA mutants and to really generate a ghost BRCA wild type, you can lose the cancer cell can lose all the sensitivities to PARP inhibitor. So that's known, right? So that's a big percentage of the patients that are doing that. So eventually, all patients will relapse almost, right? So that just speaking about the job is not done here, right? So that's why I think you can see many companies are working on additional HR deficient synthetic lethal target, right? Including POQ, we are working on USP1, and hopefully those target with different mechanism, right? That, for example, some of the target, they may work even in the BRCA revertant way, right? The synthetic lethal mechanism might be different from PARP inhibitor. In that way, you may be able to, for example, treating those patients after relapse or even upfront, do your combination to extend the potential, benefit there. So I don't know whether that answers your question.
Thank you very much. Thank you.
Can I add something? But not. I was first thinking about throwing brick and getting some jade from Alan, but I'm going through that anyway. So I think there are great questions for the PPAR. I read from paper. I did not do this program myself, but they start called the BRCA, right? They could expand, right? Some also similar to BRCA mutation 1, mutation 2, and so they're going to expand the patient population. That's right, number one, that they could do that. And secondly, for the second question for the resistance, So they could, like, I think Alan already mentioned, they could use other, right? Because why they have resistance? Because it's escape, right? I mean, their other target, other protein can repair the DSB, right? The double strand break, right? So you can do the combination like ATR, right? Some other proteins in the DDR pathway. And then so you can, I mean, it's a like cliche combination therapy. Probably in the end, you know, you have to go through the combination therapy, even with some immune oncology targets, right? Currently, so many clinical trials by combining PARP1, ATR, you know, inhibitors. And I want to add one thing on top of this. I think there's a company called Oxino. That this company, that's the only compound they have the oligotide. Others all small molecules, but this oligotide they were thinking about just it's like a decoy for the DSB, and then you know so the DSB couldn't repair, you know, the damaged DNA couldn't repair. It's like decoyed, and so they let it you know, it's like cover multiple proteins. So that's one method that people are working on. But of course, we don't know the toxicity yet until we get the phase 2 POC data for that. I just want to add that. Thank you.
Thank you.
I also want to pitch in and give another perspective. So Zuye, the point that you point to about the emergence of resistance to synthetic lethal therapies is of course more general. It characterizes a response to targeted therapies as a whole, right? So even very successful targeted therapies, in the vast majority of the cases, resistance emerges after some time, but you buy time, and that's very valuable, and so on and so on. So, you know, perhaps the only, or one of the only types of therapy where, aside of surgery, of course, where we see complete remission and response is immunotherapy, right? With checkpoint therapy and cellular therapy, but regrettably, that's still in only a subset of cancer types and only in a limited fraction of the patients and so on. But you see durable responses, right? So, and it's, when you think about it, with targeted therapies, you're basically selecting resistant clones, right? Because most tumors are heterogeneous, and there is intratumor heterogeneity, and that's of course a challenge. So the response as a community that we have is that, okay, we look for combinations, right, of targeted therapies and targeted therapies with other modalities, with checkpoint therapies. But that's a complex issue, right? Because while you scale the efficacy, you also scale the toxicity. So in short, these are very serious challenges. So, for example, the NCI now, in addition to the MATCH program, is coming out with a new flagship program, which is Combo Match, and which will test quite exhaustively different combinations, promising combinations in clinical trials, which the NCI will fund, and so on and so on. But remains to be seen, right? The foe is very serious. I just want to say that synthetic lethality actually offers an interesting opportunity to try and fight intratumor heterogeneity. Because suppose you find a target or a drug which is a hub, which has many synthetic lethal partners. And suppose you treat a patient with such a drug that targets such a synthetic lethal hub, then at least in theory, conceptually, you have a chance to kill many clones, many birds in one stroke. And why is that? Because this target has many different synthetic lethal partners. Each of them may be inactivated in different clones, and you're getting an opportunity to try and fight heterogeneity. This looks, you know, interesting conceptually, but we still need to study carefully if and to what level it works. Okay? So we haven't solved the problem of tumor heterogeneity and resistance. That's probably the biggest single challenge that we have in cancer therapy. Okay, and we have a lot of buzzwords, and combinations is one such, but step by step, it's a very serious challenge. If I may just ask, I'm not the moderator of the discussion.
Oh, go ahead, go ahead.
No, I just want to ask people who feel comfortable with, if they would like to, open their cameras, and so we can see each other. We are all human beings, of course, only if you are comfortable, because I feel a bit bizarre in this virtual world, talking to names without seeing the people. I'm as Ning said, I'm an MD PhD. I'm actually a psychiatrist in my residency, which I never. So I'm interested in people. So anyone who feels comfortable and wants to show himself, that would be awesome, and you will enjoy the discussion much more. But only if you're, of course, okay with that. And then we can see you while you ask questions and relate to you and so on. So please shoot.
That's great. Yeah, we got one more like Jiang Yu, like raise his hands. Like, go ahead. Yeah.
Thank you, Ning. Thank you for picking my question. Thanks, Dr. Robin and Dr. Beck answer my question. Appreciate that. So yeah, this is Jiang Yu from St. Jude. I'm doing computational biology. The reason I'm asking that question because we're exactly dealing with that open clinical trial, try to use RNA-seq based kind of biomarker to guide the treatment. We got a couple of stories recently published as well. By the way, I was trained with
what type of treatments? What type of treatments were you?
Leukemia. So St. Jude, we're treating T-cell leukemia, TIL, which is
Can you please kindly send me these papers and your work?
Oh,
I would love to see.
Absolutely, it was a Nature Cancer paper a couple months ago. So
okay, in my ignorance, I didn't see.
No. Of course not.
Right.
Yeah. Thank you for that. So to continue, we were fascinated by your cell paper on select algorithm. So agent. So one question about that is, you did not mention that. So what's your thoughts or how useful the phylogenetic evidence you put in that paper? When you derive the SL score, you put phylogenetic screen or analysis. Wonder how useful that in the final executive score.
It is very useful. It is very powerful, actually. And actually, we have an earlier publication on an earlier method called IL. You can look it up in Nature Communications 2018, but you'll see there is a specific figure of the contribution of each step of this 4-step algorithm and so on to the accuracy of the identification of the synthetic lethal partners and so on, and the overall accuracy. But we believe that this is a very important step.
Great, that's good to know that. Yeah, this was first time we we never thought about that, so it's good good to know that. Cool, thank you.
Great. Yeah, any audience want to ask question, please raise your hand. I mean, while we are discussing, some of the audience also suggested whether any of our speakers think there might be some interesting questions like you guys want to discuss and also want to ask to some of the other speakers. Maybe that's a very interesting setup. Like I just want to see whether any of our speakers want to ask any question to the other speakers or want to discuss any questions particularly.
Can I start?
Sure, yeah, go ahead, Yuan.
Yeah, sure. I do have a question for Alan about the immuno new target identification. I don't know how much you can talk about it, right? But I think currently part one, I think they have a combination with the PD-1, right? There's some checkpoint inhibitors. But other than that, I don't see any other IO targets that are doing combination clinical trials with PARP ATR. I just wondering for the Gilead collaboration, how much can you comment how you do that and where you are I'm not sure how much you can disclose, but I could not find any publications for that portion.
Yeah, absolutely. I think at a high level, I can definitely comment. I think the whole concept of combining PARP inhibitor, ATR, or the other DDR target has the rationale of inducing potentially interferon or other immune response, and then that will enhancing the immune therapy efficacy. So the concept we are bringing forward are actually different. We're not a DDR company, and we are doing de novo target discovery. So what we are trying to work on are, for example, we already know STK11 knockout has a very profound immune suppressive effect, right? So, and we know this is cancer cell intrinsic because in the same isogenic model you put in mice, right? If you lose STK11, that effect will be there. So, the type of discovery we're running is really, using the CRISPR tool in vivo, then looking for nodes that you can knock out that can reverse this STK11 effect. And that will be your combination partner with any of the immune reagent, whatever you're using in this experiment. So it's not restricted to the, the listed synthetic lethal target, particularly in the DDR field. So it's a very different concept.
Okay, thank you.
Great. Any other questions like the speakers want to bring up while our audience are thinking about their questions? Okay. I think we got Previn. Sorry, I'm not sure whether I pronounced it correctly. Yeah, I will unmute you. Yeah, go ahead. Yeah.
Yeah. So this is Praveen. I'm the Director of Business Development at Incubator in Houston. I've been following the field, very interesting, and thanks for the competitive landscape that you have provided. So this is in respect to the targets. I'm trying to learn a little bit more regarding the targets. For example, you have the PRMT5, and there are multiple targets, the MTAP deletion, PRMT5, and then there is pol theta. So I was wondering, do you have any insights into if one of the targets might work better in solid tumors versus others? So that would be great, because I'm looking for, I think the data is going to come up for IDEAYA Therapeutics, as you have mentioned, in the second half of this year, the initial data. But I was just wondering whether if you have any insights into any of these specific targets for solid tumors, that would be great. Thank you.
All right, I guess that question is directed at me. So first of all, I think there are basically two targets, right? MAT2A inhibitor and PRMT5 inhibitor in the MTAP loss field, right? So both of them were discovered through those the drive and also Achilles shRNA screen. The one thing I just want to emphasize is that most of the existing PRMT5 inhibitors, including GSK, Prelude, and J&J, those are the inhibitors that are developed, or either, I guess Prelude is after, but many of them are developed before that synthetic lethal knowledge is out there. And the inhibitor now later on, what we figure out is they're targeting the PRMT5 SAM complex, which is SAM is a natural cofactor of PRMT5. But what you really want to target is PRMT5 MTA complex, which is become abundant once you lose MTAP in that cell. So those are very fundamentally very different inhibitors. The SAM targeting inhibitors, they will not show any synthetic lethality there. So that's one I just want to emphasize. Then, between PRMT5 and MAT2A, this may sound like bashing, that type of thing, but I think what we have considered between those two targets are, in terms of the mechanism, right? MAT2A basically achieved the synthetic lethality by lowering the SAM level in the cell, right? The analogy I often use is, this is given that SAM is such an important cofactor for many methyltransferase, right? Targeting SAM, lowering SAM in the cell to inhibit a methyltransferase is almost like trying to target A kinase by lowering ATP level. Okay, so there is a floor in terms of how much you can really lower the SAM level, right? Which are that you can figure out from the data from Agios and IDEA. So I think naturally there is a narrower therapeutic window. I do think the inhibitor can produce selective kill, but the window between the mutant versus wild type is much smaller. So and we consider that actually at 30 days for choosing the program. And we think from the biology side, PRMT5, a true MTA cooperative PRMT5 inhibitor is really the best choice in this case.
Great. Thanks very much for your thoughts. Can you give me any insights into pole theta? I was really intrigued by the target, and if you can provide any insights, that will be great.
Anything I provide will sound like a bashing at this point. No.
No, that's great to hear your perspective. Sorry, yeah, I'm not looking at, I was just intrigued by the targets and multiple companies pursuing. So I just want to get your personal thoughts. Not on behalf of Tango.
Yeah. No, I think I think it's a valid target. I think it's a definitely we see the target came out from our screen as well. Although I have to say, the spectrum of where those targets can work, right? We have some targets that disclose or not disclose. What we find is heterogeneity in terms of the response, even in the same HR deficient setting, right? So there are a few cell lines that are particularly sensitive to pole theta knockout, and other target has their favorite spectrum. So there are complexities in terms of where they will work. So I think it's at this point, it's very hard to compare to say which target is good or, versus others.
Sure.
But just to say, I do think it's a very valid target.
Sure. Yeah. Great to hear. And I also follow another company, which is RTOs Therapeutics, which you might be very familiar with. In terms of the targets, again, DDR, some of the same ones that you have mentioned. That's great.
Great. I think I have a question. I feel like Alan raised a very interesting question. I didn't think before, but now I'm very curious about, which is like, Alan, you mentioned, even though some of the genetic background context, they are similar, but they still respond very heterogeneously. So therefore, I wonder whether there are some factors beyond of genetic context or background. Of course, we know immune component is one of the important component, but other than that, whether other factors we should also consider when we push this into precision oncology.
Yeah, I think in my mind there's no doubt, right? So I think this is what Ethan was also talking about, right? Looking for multiple factors that driving, and this is actually the reason when sometimes we do the screen, we want to use a clean system, which will give you exactly the answer you want. And, then using the cell line to confirm whether do you see that. I think the heterogeneity of the real world cancer cells, right? That sometimes will kill you, right? Will really shadow the real signal there. So I think, but to figure out exactly what is driving those heterogeneity, I think it's not a small task. Those are very, very challenging.
I also think that Praveen asked, how do we know in which cancer types, which genomic context will different synthetic lethal therapies may be more likely to work? So this is one of the places where actually computational analysis can help, because of course you can't screen all genomic context and all the cell lines and all the King sources and all King expands, and instead you can look at large, patients' cancer collections and try to understand that. And I think Pangea is doing quite a bit of work in this direction. Tuvik, maybe you want to say something about that.
Sure. So yeah, that's actually our main focus, looking at patient omics and trying to figure out the most suitable treatments. And it relates to a couple of questions before. I wanted to mention, well, there are a couple of points here. First, I think we need to look beyond just synthetic lethality. That's perhaps the most well-known type of genetic interaction, but it's not the only one. And in particular, synthetic rescue is a very important type of interaction, and we see its importance in predicting.
Maybe you can explain what is synthetic, because it's much less known than synthetic lethality. Maybe you can briefly explain.
Sure. So well, maybe you can explain, because you're
no.
On that topic.
No, go ahead. Really, I enjoy listening to you.
So basically, in synthetic rescue, we are talking about a type of interaction where one gene inactivation is in fact sufficient to induce lethality, but then another gene can come in and rescue the cell, either by upregulation or downregulation. Okay, so whereas in synthetic lethality it takes two to lead to cell death.
Takes two to tango therapeutics. Synthetic lethality takes two to tango therapeutics. Right, please, don't let me disturb you, Tuvik. Please go ahead.
Please go ahead. So in synthetic rescue, one is enough to kill, and the other gene comes in and rescues the cell. And in Pangea, we're actually looking at networks of genetic interactions which comprise both synthetic lethal interactions and synthetic rescue interactions. And we find that such combined networks are very effective in predicting a patient response. And there was one particular case of a patient that we analyzed, and that patient came in with a rare cancer, and she was a young woman, underwent several lines of treatment, and underwent also a comprehensive genomic profiling, which brought up no actionable mutations. And in particular, she was found not to be eligible for immunotherapy. So standard biomarkers, microsatellite instability, and tumor mutational burden, everything indicated that she would not respond to immunotherapy, to immune checkpoint blockade. And in contrast, our analysis of the synthetic lethal and synthetic rescue environment of the checkpoint genes indicated that she is likely to respond to combination immunotherapy. And her physician, having tried everything else, decided to go with that. And it's been almost a year ago, and the patient is currently in complete response. So both the original tumor and the metastasis have all but disappeared. So I think it testifies to the strength of looking beyond just one pair, looking at multiple interactions and multiple types of interactions. So I think there's hope.
That's great. Very nice discussion. Like, just want to check whether any audience want to ask questions. Please raise your hand. While the audience are thinking about their questions, maybe I could add. Oh yeah, Frank, yeah, go ahead. Yeah.
Hi, my name is Frank. I'm a geneticist by training as well. I used to be working in a lab that works on synthetic lethality. I have a couple of questions here. First of all, I think early on, Alan mentioned that a lot of the synthetic lethality targets currently in those companies are working on are focused on, for example, DDR targets. Maybe this is due to the fact that PARP inhibitor became so popular in the past few years. But despite the fact that, or despite PARP inhibitors, other targets, for example, POLQ or WRN1 targets are also being developed for a couple of years, but we haven't seen any of them coming out, like say in the clinic or with a really good results, or none of them have been approved by FDA or any of those healthcare authorities. So do you have any thought on why these targets that were so promising at preclinical data, and then they found that they are also targeting a lot of synthetic lethal hubs in the cancer cells, and they're not coming out yet? But like my personal opinion on, for example, PARP inhibitors is not the fact that it's just target PARP, but it's more of it's being a PARP trapper, so that it has a more effect on just inhibiting PARP. So do you think maybe POLQ or WRN1, these DDR genes that are clinging onto the DNA, and maybe those inhibitors targeting those proteins have to have a trapping characteristics to make it work? Just want to see your opinions on these.
So the second target you mentioned is Warner. So other than
WRN,
right,
WRN1.
Warner. Yes, yeah.
Well, yes. Yep.
Yeah. So, maybe I can comment first. I think first of all, development drug takes a long time. If you look at how long has AZ been developing olaparib, you will probably get some feeling, right? Obviously, they made some mistake early days for the strategy, but as far as I know, for example, POLQ is actually very challenging target, right? Repair even from outside, I see that they have the pipeline goes forward, then come back and goes forward again. So you can imagine it's actually challenging. I don't think that target has seen the patient yet, so it's very early to say, right? Warner, I actually do think that this is a very, very exciting target for not for HRD, but for the MSI microsatellite instability. That target, I think it's also a very challenging target also. So part of that is drug discovery. I think what you mentioned about the trapping activity, that concern is certainly there. I think, but different targets are different. So we will have to see things play out there. So I think it's too early to say, right, whether this is, the new batch of HRD and or synthetic lethal target is working, right? Then you have your probably third group where you didn't mention, right? Those are ATR, V1, CHEK1. Those I think, to me, the challenges are, those are the target with narrow therapeutic window. Those reagents are very toxic to the normal cells as well. And also to me, the synthetic lethal hypothesis is not as clear, right, to what patient to select. So I think that the path for developing those reagents, in my mind, is going to be more challenging.
I can add a couple, one point on to that. I think that's a good question, right? But now currently, right, at least 18 drugs in the earlier phase 1, phase 2, right? So if we can get a good readout of, POC readout in the phase 2, that will be the inflection point that I think the investment will come even more than current situation. For example, ATR has some the Bayer compound has some promising phase 1 result, like the partial response about, I think 10 patients, maybe around 30%. I think the overall response rate. but still it's in the phase 1, and I have not seen the safety data. You know, as Alan mentioned, that they have narrow window. So I think if they have more patients, if they can do a pivotal registration trial in phase 2, if they can see good signal, I think that will be an inflection point for the whole for the synthetic lethality field, right? Currently only PARP, and it has trapping capabilities, but I think other targets just need a little bit time to tell.
So I have a question. May I?
Go ahead. Go ahead.
Ellen, I apologize. I jumped into the discussion a little later. My name is Ronai, and I've been interested in PMRT5 over the last few years. And one of our studies demonstrated how PMRT5 inhibitor can overcome immune evasion and therefore can be used to convert cold tumor to hot tumor. That was about a year ago. So I just caught that you at one point in passing mentioned PMRT5 together with MTA as a combination. I may have missed it. Can you expand why? Originally, PMRT5 was identified as synthetic lethal with MTA. That was 2 science papers about 3 or 4 years ago. But I'm not sure that MTA still holds in the same way, or we understand it today as we understood it 3 or 4 years ago. Can you elaborate on that, and what would be a marker that you can use to trace the efficiency of such combination of PMRT5 inhibition?
Yeah, sure. This is a very complicated story because a lot of the gene names are very similar, so it's very easy to get confused, right? So the PRMT5 cofactor is SAM, right, which is activating cofactor because it can transfer methyl group. MTA is a SAM derivative, right? It looks similar to SAM, but binding to the same binding pocket, but cannot really activate the enzyme. So essentially, MTA bind PRMT5 is inactive. So MTA is usually metabolized very quickly by the enzyme called MTAP. So those are the confusing piece of a couple of the things. MTAP is lost on the chromosome 9q together with many things. So it's a very large population. So co-deleted usually with CDK and 2A. Therefore, it's about 10% of all solid tumors. So in this
30% or 40% in PDAC, in pediatric cancer.
Yes. Yeah. So and when MTAP is lost, so MTA will accumulate and goes very high. And so in those settings, MTA PRMT5 is a dominant complex in the cell. In the normal cell, SAM PRMT5 complex is the dominant form. Okay, so essentially, it will distill down to making an inhibitor targeting the PRMT5 MTA complex versus MTA PRMT5 SAM complex. So that's really the distinction between all the existing PRMT5 inhibitor and the type of inhibitor we are developing. So as a result, as I can tell you, the SAM-based PRMT5 inhibitor, they look, if you test them against a panel cell line, they look pan-lethal. Okay, because PRMT5 is essential part of a lot of essential functions. With our inhibitor, we can see clearly it only hits the MTAP loss cell lines. Okay, so there is a selectivity built in right there. And
so some of the concern has been that MTA does not selectively only target PMRT5, it has additional target to what was originally thought. Yeah. So the question is, how stable is the complex between MTAP, MTA, and PMRT5? Is it a solid complex or transitionally one?
Yeah. Interestingly, although there are many other 30 or 40 other methyltransferases, right? The affinity of MTA to the enzyme, to other transferases are much, much lower compared to PRMT5. PRMT5 is an outlier. So in our hand, I think the affinity there is probably, if not anything equivalent to SAM. So, you know, that's why in the genetic screen, you actually can see PRMT5 dropout, but none with other methyltransferase dropout. So yeah, so that's I think in a nutshell how we are thinking about. So I think to your question, I think this is a very stable complex. Okay.
Great.
And, can be targeted.
Yeah, I think thanks, Alan, for really comprehensive and nice explanation. I guess this might be a little bit too specific to some of the audience, and also I think you know, this really really fruitful and informative discussion, and we are just about to close. So in the end, I just you know, want to have some fun stuff, which is like I hope I could have each of our speakers to comment on what do you think might be the most important challenge or questions we need to address in synthetic lethality field in the next 5 or 10 years, and some of your thoughts, like how we could potentially do that. I think that's really something fun, like many audience want to hear from you, and either in scientific side or of course, and also in the business side. So I just want to hear you in the very end.
Sure, guys, go ahead. Unless if nobody, I will. But please go ahead. Anyone?
I can start. Ethan, you know, I can start. So I think the most challenging for me, you know, the earlier new target identification, you know, how reliable the pair, you know, identification, how robust that is. I think that's still challenging. But as Ethan mentioned, I think the single cell, you know, transcriptome might be one option. I borrowed from Ethan. Others, I think just with the refinement of CRISPR technology, with time, with so many groups working on it, I think that will improve automatically. That's all from me.
Great,
Thank
thanks.
you. Alan, do you want to?
Yeah, no, this is, I think, a complicated question. Maybe I can answer by saying, you know, that one, I do agree that, you know, the target is really the key, right? So, you know, I'm stealing, you know, the chairman of our board, Alexis Borges, or he was always looking for the glorious target, meaning that target is truly selective, highly effective, and the effect size is big, right? So you know, and in now a couple years down the line, I think we know what kind of target we're looking for, right? Especially even at the genetic stage, we want a very strong in vivo genetic validation, right? So those are I think will still be the challenge, both technically and also from the discovery point of view. But that's the type of target it will take to really give you the strong clinical response.
Tuvik?
So on the precision oncology front, I'm betting on liquid biopsies, and in particular transcriptomics of liquid biopsies as the next big change. And we are in active collaboration on that, and it is a very challenging field. It's really hard to get reliable data from RNA-seq in circulating tumor cells, but it is doable, and I would expect the technology to really advance in the next few years. And I think we should all be there to take advantage of that when that happens, because it would really change things if you're able to follow the patient over the course of multiple treatment cycles and over the weeks, and you know, deal with that heterogeneity that Aidan mentioned earlier as one of the big detriments of effective treatment, and find the next good drug to treat the patient with. I think that would really revolutionize the way we treat patients in precision oncology.
So thank you, Tuvik. Thank you, everyone. I will just give you my perspective. So we are working on synthetic lethality, the computational identification of since 2012, 2013. So it's a very long range project in our lab. In fact, we published our first synthetic lethal identification pipeline in Cell in 2014. It was called DAISY. And we have gone a long way since then, and hopefully, I think we have considerably improved along the way, and we are learning, but we are still not, there's a lot to be done. So I think one major challenge for my lab is to continue and improve our approaches, and we are working very hard now to infer synthetic lethal partners from single cell data. And once we will be able to do that, that will be transformative, because we will think about it. Each patient samples give you thousands of samples, okay? And you can identify genetic interactions between a gene in a tumor cell and another gene in a CD8 cell or CD4 cell, okay? So currently we are not yet succeeding to do it. There's a lot of noise, but guys, we will, okay? And we will do it in a matter of a year or so. That's what I also said last year, right? But whatever, we will do it, okay? The second thing is that we have big projects aiming to harness in different types of genetic interactions to identify combinations with checkpoint therapy. Remains to be seen. We are working on that, and there is ways to validate and test that. And lastly, I want to say, in the spirit of this discussion, we had experimental people and computational people. I think that synthetic lethality is a wonderful place where these two approaches can work together and complement each other. So, for example, I see a lot of ways in which technologies like Pangea can work together with and boost these amazing synthetic lethality CRISPR companies like Tango are doing. And that's a potential, for wonderful synergism between experimental and computational work. So let's try and change the world together. That's what, if we can do even just a bit.
That's great. I mean, it's really inspirational discussion, and it's really something wonderful for us, for the audience, to hear the leaders' thoughts. And we are definitely looking forward for the chance for Tango and for Pangea to work together for the computational side and experimental side, CRISPR, single cell, IVD, all those things together. And of course, all we want to do is trying to kill tumor, to cure the patients, and want to improve people's life. And I think that's the one goal we all want to do, and we definitely want to do it together in different ways.
Here, here, absolutely, anything. That's the NCI mission. That's why I get up in the morning, and work as a federal clerk in our institution, right? Together with amazing collaborators. I see Dr. Aldape here. I'm so fortunate to work with such people. Guys, you have no idea. Yeah.
Great. I mean, it's really wonderful discussions. I know we already passed the time, but I'm really still very excited about all we discussed today, and I even can see the future now. And I know some of the audience may want to follow up with the speakers, so just feel free to just on your way to email Chance, follow up. I don't know, you may find your own way, but I hope this discussion can inspire different thoughts in this field, and of course strengthen some of the collaborations. And we definitely looking forward to hear more news from every of the speakers. And we wish, we have the one goal together. By doing that, I hope I can just conclude today's discussion. And I want to thank again for our speakers' time and the really inspirational discussion, and for our audience, for our organizers, for your time. And we are looking for your all the news from you. Yeah.
Thank you so much,
Okay.
and thank you, Ning Wang.
Thank you for your invitation.
Yeah, thank you. It's nice. It's
Okay.
pleasure to meet all of you. Thank you. Bye.
Okay,
Bye-bye, guys. Have a nice weekend.
You too. Bye.
Thank you.
Yeah.
Yeah.